Mind-NIMHANS. So on behalf of my co-organizers, I welcome all of you to the second installment of our webinar series at the Center of Brain and Mind. The Center of Brain and Mind, for those of you who are joining us for the first time, is a joint collaboration between investigators at NIMHANS and the National Center for Biological Sciences at Bangalore, with the objective of employing multi-pronged approaches into studying severe mental illness using sophisticated clinical investigations, modern human genetics, and stem cell technology. So to introduce today's speaker, I'll now turn it over to Dr. Ritika Sood, who is a senior lab scientist at the Center for Brain and Mind and coordinator of the lab at NIMHANS, the Center for Brain and Mind. Yes, Ritika.
Thank you. I'm very excited to welcome all of you. Today, we are joined by Dr. Jens Hjerling-Leffler, who is an associate professor and principal investigator in the Department of Medical Biochemistry and Biophysics at the Karolinska Institute. He describes himself as a neuroscientist navigating the treacherous waters between molecules, physiology, and disease. His research group is interested in the genetic and cellular mechanisms of postnatal and adolescent maturation and its role in psychiatric disorders. They apply modern molecular methods to probe cellular changes and the function of individual cell types in the brain. Today, he will speak to us on how single-cell sequencing is helping unravel the mysteries in schizophrenia. Before I turn it over to him, just a couple quick logistical things. As and when you have questions, please type them in the chat box. We'll take them at the end of the talk. Jens, over to you.
Okay, great. Thank you, Ritika and Bijou, for this kind invitation. I'm really excited to share the data and to be this as a formal start of our relationship. So so today I'm going to talk about the efforts that we're having in our lab. And one of the things that we're trying to do is to try to understand where in the brain we should be looking for disease-related changes or causative changes for different disorders. And we focus on schizophrenia. Oh, no, it's not working. Oh, no, here you go. Which is a mainly genetic disorder with a high heritability. It's pretty common across the globe. Between 0.5% to 1% of people are affected. And this is largely remains the same across different kind of cultures. These numbers are not drastically changed by wars, famine, or pandemics, which is in of itself a really interesting feature. It also means it's a huge unmet medical need. We have very limited treatments. The personal suffering for patients and their families is huge, and also it's a very large cost to society, mainly due to loss of income, because it affects people early on in life, and then they live with the disease for their whole life. So down here in the circle plots, I've been trying to describe a simplified version of the genetic architecture of different disorders. So first you have severe intellectual disability, autism spectrum disorder, and schizophrenia. And the different colors denotes different types of mutations that that are associated with these disorders. And as you can see, intellectual disability and autism has a much larger fraction of cases that can be attributed to known genetic causes. This means that there's rare mutations, or not so rare in certain cases, that we can clinically investigate. And also these kind of mutations gives us really good handles experimentally to build mouse models or to look for these kind of changes in experimentally. So for schizophrenia, this is not the case. The gray here means mainly unknown genetic causes, and in this case, it's SNP heritability is a large proportion of this, which means that there are thousands of genes, each with small effect sizes, that contributes to this disorder, and very few cases can be ascribed to known large mutations, and these have classically included the copy number variation mutations, such as 22q11 maybe being the one most famous one. Recently, last year, a few rare protein truncating or loss of function mutations was found with large effect size, but they are exceedingly rare, making up less than 1% of cases that can be attributed to these kinds of de novo mutations. So this means that experimental experimental design and investigating schizophrenia is a very hard problem. So this has led to one of the biggest problems that I think we have, which is that large-scale industry has been divesting due to high risk. So I actually went around and I interviewed a bunch of leading people within Big Pharma and asked them, what would you need to come back to the table? Because the idea was that if I, you know, through the funding that I have, the small funding that I have in my lab, if I can contribute to making the playing field easier and bring Big Pharma investment back into schizophrenia research, I would have a great leverage on our efforts and the money that we have. So, and the things that they told us then is that their main problem is that we have such a bad understanding of the underlying biology. That's true. And, you know, the other thing that they had a problem is that they, you know, we put a lot of money into genetics, and unfortunately, genetics have failed to deliver on targets. It didn't tell us which genes. There's no such thing as a schizophrenia gene that came out of the genetics. The genetics has, in my mind, been hugely successful in terms of really describing the genetic architecture of schizophrenia. But unfortunately, it turned out that the image is very complex, and there's no such thing as the schizophrenia gene that we could target. This has led to a massive wasted neuroscience effort. So there are so many big papers. They're great papers, great endeavors into understanding what biology lies behind, like what kind of neurobiology lies behind the function of different genes. Problem was that the connection to clinics or connection to cases was really weak, because many of them focused on these early findings where we thought we would have found the schizophrenia genes that then later on turned out to have a very low effect size. So a lot of great biology, neurobiology has come out of this, but the relevance to the disorder is highly questionable. And then the fourth thing that they mentioned, which they have a problem, is patient stratification. That is basically, if you want to run a clinical trial, if you include all cases that have been diagnosed with schizophrenia, if that's your only criteria, the risk is that only a subset of those patients respond to your treatment, which means that you would have a diluted statistical effects in your clinical trial, and therefore having much harder problems trying to identifying drugs. So this leads, you know, as a scientist, you know, big problems means that for us, it's a big opportunity. And there's a few questions that we're really interested in. One is if schizophrenia might be a brain state that is reachable by several different avenues. That's saying, like, maybe it's a common state that the mind can go to, but you can reach that state by several different mechanisms. Question is also whether we can go from the genetics to, with all these thousands of risk genes, to determine molecular signatures or determine cell types or molecular signatures where they converge. And those could be potential drug targets. One thing is also the developmental time points, where schizophrenia risk acts. Is it mainly during your embryology, or is it later on? And around adolescence when the disease manifests. And then, of course, the stratification. Is schizophrenia really one disorder? And this is a point relates to the first point. You know, is it one state? Is it several different states? And if it is one state, is it possible that you can have many underlying kind of perturbations that leads toward that state or not? So this is where we're trying to make a difference in my lab. And I think that the challenge that we have here, the challenges that we face is diagnosis. This is basically for the different types of patient material that we can access, for GWASs, etc. The diagnosis is key. The better and more detailed this is, the better description we have, the better you know, we can do far away or afterwards. We also have the disease diversity. We don't know. There's a lot of comorbidities in schizophrenia, and we don't know how that really plays out in our data. A major problem is the scale of studies, because you know, we moved from very small scale. It's kind of like medium scale. I think what I'm going to talk about today is, I would still say it's small scale, but it's definitely larger than it was a few years ago. But with human diversity and disease diversity on top of that, I think that we need to reach the scale of thousands of patients or thousands of samples also for molecular studies, not just for genetic studies. And then the major problem is, of course, disease modeling. How can we model something that we don't understand? We don't have clear genetic disruptions that we can model. We can model polygenicity in IPS studies. Today, the problem is maybe that they are very expensive experiments. So the throughput there, you would like to have thousands of patients or thousands different lines there too, but that's unattainable today. Okay, so these are what we're trying to achieve and the challenges that we have ahead. So one thing that I'm interested in is to see whether we can have a data-driven kind of approach to schizophrenia biology. And I see there's mainly three ways or three different data modalities that will give us a chance to address this. One is, of course, the clinical heterogeneity. This is the interaction with doctors, and this is very large efforts across the world is going into this, trying to harmonize how we diagnose and what we diagnose and what kind of data goes into diagnosis, etc. And this is a huge ongoing effort, which I am not taking part of at all. The other two modalities that we can do screening in patients is one, we can take a blood sample and do the genetics. This is massive efforts. We now have several tens of thousands in our GWAS data. And the third one, I think that what we could do easily in the patients is, of course, do brain scanning and brain imaging of the patients. These are non-invasive methods. So these three different modalities, if we can find that they point towards the same kind of biology, or if they agree with each other, at least we're starting to get a handle on this kind of gray cloud that is schizophrenia. Sorry, I'm trying to, here we go. So I mentioned this, the architecture of schizophrenia, right? And this plot here on the left is from a paper published last year. In blue, you can see the common variants. This is all the gray here explains the 95% of the cases of patients that we know of. And so this is the vast majority of cases. They only have mutations that have very small effect size and that are not being deleted from the population. And this alone attributes to 95% of cases. Then we have the really rare type of mutations, and they have very large effect sizes. And you can see that the copy number variations, the CNVs, are classically being studied, and there's a number of them that increase your risk for schizophrenia. And then there's the newly identified protein truncating variants or loss of function variants that also have a large effect size on your risk for schizophrenia. But the problem is with these, with the most of the cases being derived from common variants, is that we have a really hard time finding structure in this. They are mainly additive. It seems like each point gives, but we don't know how to find the structure here. What's been known, you can do gene set analysis, and what's been shown is that these are highly enriched for synaptic genes that have a synaptic function. So our question was whether we can use biology to structure genetic data. So basically say we have all these thousands of risk genes. Can we use some kind of biological measure to try to understand where these genes are acting? And one of these things, fundamental way to look at gene expression in the brain is really in cell types. So we can ask the question, all of these risk genes, which cell types are using these risk genes? And the assumption then would be that if you're not using risk genes, that cell type in itself is probably not involved in the etiology of the disorder. If you, on the other hand, are using a lot of risk genes, the chances are that you are more perturbed at some point and therefore contribute to the risk state more. And if you look at the literature, all cell types in the central nervous system has at some point been implicated in schizophrenia, because we've gone the other way around, right? We've said, I'm interested in astrocytes, let's look at schizophrenia. Lo and behold, there are astrocytic changes. So these are really important. And then you can do that for each of the cell types that are there. But the question is, can we look can we go in without having these notions where to look for the signal, and instead use a data-driven approach? And to do this, we integrate GWAS findings with single-cell RNA sequencing. My lab was early on pioneering single-cell sequencing in the brain, so we had access to really to big data sets and from mouse. And so what we basically did is we took the single-cell data set, and for each cell type, and I'm showing the example here of a median spiny neuron from the striatum of the mouse. For each cell type, we calculated a gene expression, gene specificity matrix. So in the colors here are different genes and their density. This is a log scale axis. But in the first ones here, these are the most not specific genes. So in green, basically, this is everything you need to make a cell. This is the mitochondria, the nucleus, the cell membrane, you know, the stuff that you need that every cell in the brain has in common. As you move up in specificity, you're starting to see genes that are now more and more specific. So in orange here, you have this and in the orange colors, you have the genes that are necessary to be a neuron. So basically, these are genes that are shared between neurons, roughly 50% of the brain. And as you move up in specificity, you are getting more and more specific genes. And these genes are now typically those genes that we think are defining the function of a neuron. Its connectivity, its firing properties, its response to receptors. As an example, I put in the dopamine receptor here, which is quite specific for median spiny neurons. It's expressed in other cell types, but mainly in these cells. And that's the example here. So now we have this specificity matrix. We can now divide that into deciles. That's the different colors that you're seeing. And we can ask the question, if we use LD score regression to look for heritability enrichment in each of these deciles. And as you can see here on the right, as you move up in specificity in a median spiny neuron, you actually get higher and higher signal for schizophrenia heritability. If you then take a GWAS for height, of course, we don't think that the striatum regulates how tall you are. And as you can see, this line is flat. So as you go up, you are actually decreasing the heritability for height in your sample, in your data. So we've done a lot of work on trying to figure out these kind of methods. And the method that we ended up with is actually to look at the top decile. We looked at the slope, we changed a lot of different parameters, but the most stable entity that we can look at is actually the enrichment in the top decile. And so that's what we're doing nowadays. So we showed this in a mouse, we showed that this is highly enriched in neurons, certain subclasses of neurons, etc. But that was always in a mouse. But a few months ago, there was a new data set published in Science. It says bioRxiv on this slide, but that's last year when it came out. But it was published in Science a few weeks ago. So here, Sten Linnarsson and colleagues from the Allen Brain Institute, they had taken 4 different human brains, and they had performed 106 different dissections from different brain areas, and then performed single nuclei RNA-seq on this. So this is the first time that we have a very complete map, or not complete, but a very broad map of all cell types present in the human brain. And this is what you see on the right. This is a UMAP clustering. I think maybe you've seen this before, but this is basically each dot in this graph is a cell, and if 2 dots are close to each other, they are very similar to each other. And this is used to visualize cell types in single cell data sets. And you can see here that below the dotted line here, you have the non-neuronal cells, and then you have cortical cells, amygdala cells, median spiny neurons of the striatum, classical interneurons of the cortex and other brain areas. And then you have something called the splatter, which is basically a mix of all rare cell types. So dopaminergic neurons are in here, serotonergic neurons are in here, all these kind of like very distinct, but neurons that are in smaller proportions in the brain. Okay, so now we have this kind of map. So instead of going to the brain, to the mouse brain, we can now perform this kind of analysis in the human brain. And if we do so, this is the same map, but now we've colored it for the heritability of schizophrenia, for schizophrenia. And now we can see that there's very little over here. Actually, we didn't include the non-neuronal cells because there was no signal in any non-neuronal cell. So we just excluded them from the graph. But what we can see is that there are some hotspots here, and this is kind of surprising to us. We can see that there's very high enrichment of disease risk genes in amygdala excitatory cells, in hippocampus, and in what's something called EMSNs or eccentric MSNs. These turns out to be long range projecting inhibitor neurons, not only from the basal ganglia, but also from other subcortical structures, such as the amygdala and the entorhinal cortex, etc. So in B, this is the top cell types in terms of enrichment. And you can see that the top one is indeed amygdala excitatory cells, and then comes some hippocampal cells. And then the top non-amygdala hippocampal cell is actually upper layer intratelocephalic cells. And I'm highlighted it here because I'm going to come back to that a little bit later. I should say that this is all the hard work of a postdoc and a student in the lab, and in a collaboration with Patrick Sullivan and Lu Yi. But Xu Yang and Arvid did all this work. Okay, so now we have each cell type. We know each cell type, how enriched it is for schizophrenia risk. We can now move into those 106 brain dissections, because all the data came from 106 dissections of distinct areas of the brain. So now we can say, okay, for each of these areas, and you can see them, these are the rows, we can ask what's the relative contribution of the different cell types to each of these areas. So cell types are on the columns, and brain areas on the row. And here you can see that the cortical regions indeed contain a lot of cortical cells. There are some of these also in the amygdala and basal forebrain. And then the hippocampus is very specific, hippocampal excitatory neurons in the hippocampus. Amygdala excitatory cells, or what's been classified as that, is actually present mainly in the amygdala, but also in some paleocortex and temporal cortex. The thalamus is very distinct, and these are the excitatory neurons. So when you go to the inhibitor neurons, you see they are much more spread across different brain areas, and even more so if you move all the way to the right to the non-neuronal cells. Okay, so now, because we have each cell type relative contribution to each 106 areas, we can now calculate the enrichment, schizophrenia enrichment per area instead of just per cell type. And if you do that, this is a longer story, so I'm going to cut it short and show you this. You get the map on the left. So basically, you can see that the cortex is medium enriched. And there's a few nice things I want to point out. The fact that the cortex is quite homogeneous is not so surprising to us, because there's a lot of data showing that the general cell type composition across different cortical areas is very similar to each other. There are clear differences, but those are kind of like on a detailed level rather than the general architecture, cellular architecture. And if you open the brain and you look inside, what you can see that here's the caudate nucleus and so on. There's a little bit less enrichment than you see in the cortex, but the real hotspots of the whole brain is now in the hippocampus, and in the amygdala, a few amygdala nuclei. These are really the key points where for schizophrenia heritability. So I can say that this was a little bit surprising to us, and I'm kind of happy about this, because I wrote a large grant many years ago, and I wanted to do the amygdala and the hippocampus because they also have extensive connectivity to each other. The granting agency basically said, why are you doing amygdala hippocampus if you're doing schizophrenia? Everyone knows that schizophrenia is in the prefrontal cortex. And I said, okay, fine, I'll switch to prefrontal cortex. And then they said, okay, then you get the money. So I think that we have this thing in science where we like to have a few ideas early on, and then that kind of biases us where to go look for things. So the prefrontal cortex is it's a good starting point for schizophrenia, because we know that there is really prefrontal dysfunction in patients. We know that there's hypofrontality in imaging data. And we know that the prefrontal cortex development times well with the onset of the disorder. So there's many things pointing towards the prefrontal cortex. It's clearly involved in the disorder, but the fact that the genetic risk doesn't seem to be enriched there is really interesting. And I think it highlights something that I think that the other thing we know about schizophrenia risk genes is that they're enriched for synapses. So I think that schizophrenia is really a gene regulatory disorder and a connectivity disorder. These are not large statements because that's basically any brain disease is a connectivity disorder at some point. But I think it's really highlighted here that the PFC has there's clear dysfunction here, but it doesn't have a genetic risk. What it does have, prefrontal cortex, compared to most of the other areas of the cortex, is it has an amazing connectivity with the hippocampus and the amygdala. So it's possible that a lot of the frontal problems that we're observing in patients actually arises from a connectivity model where the inputs and outputs to the amygdala and hippocampus are disturbed. So to see whether this holds, we went and downloaded an fMRI data set. This is functional fMRI resting state from 50 patients and 50 controls. And we did something called recursive feature elimination, which means that you take your brain areas and then you ask a machine learning algorithm to say, can you please tell us the difference between a patient and a control? And then you try to optimize for that. And then you say, okay, you did this well. Which was the connectivity that contributed the least? You remove that, and then you restart the model. And you say, can you now please find the difference between cases and controls? And if you keep doing that, you're going to weave it down to a smaller and smaller network that maintains or increases your chance of really telling this difference. And if you do so, interestingly, what you end up with in the end is a core network, or not, it's not a central network for schizophrenia. And we've done this across the different data sets, but in a very statistical, rigorous way in 5 different iterations. We're using different starting points in different ways. And in all of these, what we always end up with, no matter how we run this, we always end up with this network, or very close to this network. So you can see here that it's the hippocampus on both sides, the amygdala, the left amygdala nucleus, the lateral nucleus, and the central nucleus. And then it's their connectivity to prefrontal structures and a few other cortical structures. So this is a nice example of where we get an alignment or a convergence of different types of analysis. One is genetics over transcriptomics to identify brain regions, or we use fMRI, and actually we identify the same kind of regions. So this is, I should say the fMRI field is a little bit corticocentric. So we have all these other default mode network and core network, etc. But they usually don't take these deeper structures into consideration when they do their models. So we would like to say now that we should be focusing maybe more on the hippocampus and the amygdala and the connectivity to prefrontal cortex if we want to understand the etiology or the origin of the disorder. Okay. Let me move to the next slide. So I want to switch gears a little bit, and another way to address these kind of issues is to do single-cell RNA sequencing in post-mortem data. I mean, this is a different type of analysis. It's completely different. Now single-cell RNA sequencing, of course, integrates not only gene regulation, but also, or not just the genetic information, but also epigenetic information, or like things that happen to the brain as you live. And of course, post-mortem is the end of life. So it is an integrated approach. It's very different from the etiology. It's more describing the phenotype or the symptomatology of the disorder. So we did this, and we ended up in the prefrontal cortex due to granting agencies. But we did 42 controls and 43 schizophrenics. We cut out the BA, or our collaborators cut out BA8, BA9 area. We did the single-cell RNA sequencing of all these patients and all their, and then you can see here the UMAP of cell types that we can deduce from this. And this is the work from Lisa Bast, Xu Yang, Fatima, and Jose Martinez Lopez in my lab. First, I want to show you a little bit of sample QC. So once you get the data, you can now, when you have this large numbers, which is really nice, you can look at how different quality metrics align, and you can see that there's a bunch of samples over here that are very similar to each other, and then we have 2 outliers, and we actually remove these outliers. That's why there's two people in grayed out in the schizophrenia cohort here. Then we have done SNP genotyping of these, so we can know their ancestry and understand their polygenic risk score, etc. And you can see most of them are of European ancestry, a few that are not. And it's nice to see that the schizophrenia genetic risk scores in patients and controls indeed were different from each other. Okay, so the first thing we asked was whether there are any change in cell type abundances across different patient and controls. And we can find no statistical difference. And you can see here that the 2 top ones is an increase in LAMP5 neurons, which is an inhibitory neurogliaform cell. We see an increase there and maybe a decrease in somatostatin neurons, but these p-values are nowhere near significance. And what I'm showing you here, these p-values are actually not FDR corrected. So they would be even higher when we correct for multiple comparison analysis. This is quite different this agrees with large-scale studies done on post-mortem materials with hundreds of participants from David Lewis. But it's complicated by another study that was published a few years ago with like I think they had like 12 or 16 cases and controls, and there they found massive changes in cell type composition. But I think as we increase this, as we increase the sample size, they are going in the same direction in that the somatostatin are lower here in their and lower in ours, and the LAMP5 ID2 here are up and up in ours. But our data shows that this is no longer statistically relevant as you increase the N's. Okay, so now we have these cell types. We can now ask the question, so what about differentially expressed genes? Which cell types have the most changes in patients and controls? So we performed differential gene expression, and we're being very conservative, actually using correct statistics, which means that you get very few genes. But you can still see that no matter what the test you perform here, you see the same kind of pattern, which is that the excitatory layer 2-3 neurons are by far the most perturbed. And if you remember, as I told you, these are also the cells that are the most enriched for genetic risk. So you're seeing that on the right-hand side over here. This is the genetic risk that we calculated based on the expression of our own data. And you see it's a really nice concordance between these 2 maps. Basically, the more risk genes for schizophrenia expressed, the larger differences you have in your phenotype. And there's a few notable differences. One is the astrocytes, which has very little genetic risk, but we actually do find differentially expressed genes, etc. Let's move. So we can look into this. So now we have we can look at the differential expression, the genetic risk for the different types of differentially expressed genes. And if we look for downregulated differentially expressed genes, they are much more enriched for schizophrenia common variants than the upregulated genes. So basically, we think that the common GWAS signal leads to, in general, as a general kind of statement, it's a downregulation. And you have upregulated genes, but those are maybe in response to the insult rather than driven by the genetics. As an opposite, if you look at these really large scale, these large effect, rare genetic variants that it drastically increases your risk of schizophrenia, you can see in this plot that green here means upregulated and red down. I'm sorry for that. It's a bit counterintuitive maybe. But what you can see that this is mainly a green area. So this is... we can say that these genes are mainly being upregulated in response to having schizophrenia, with one notable example, exception, which is CACNA1G, which is also downregulated. So this point, CACNA1G, I think, is a point of convergence between rare mutants and common variants. While I think that the rest of these, many of these common rare variants are actually protective genes. I think they have a protective role. If your brain is veering towards a schizophrenic state, your brain tries to compensate by upregulating these genes. And if you fail to do so, if you fail to upregulate them because you have one of these mutations, you are probably much more likely to fall into the schizophrenic state. But this is speculative, but that's our understanding of the data right now. Okay, so now we can look at what are these genes that are up and down regulated. If you look in the downregulated map, we can say, I would say that this is kind of mirrors the image that is seen by other labs, also from bulk sequencing, which is that there's a number of pathways that are represented in the downregulated data. We have everything from transmembrane transport, GTPase activity, mitochondria signaling, cell migration, some developmental processes, etc. On the right, you see what we call the alla medica plot, because we have a building at Karolinska that looks like this. But it's basically here, if it's red, it's down. If it's green, it's up. If it's white, it's not significant. If it's gray, it was too few to be tested. And you can see that these, a lot of these, each column is a cell type and each row is a Go term. And you can see that a lot of these programs are actually common between most cells. This is the mitochondrial process, while a few are quite specific to cell types. So here we can see basically that the RAS row small GTPase regulation is very enriched in microglia cells, but not in any other cell. And some very specific for layer 2-3 neurons change in ATP biosynthesis process. But this doesn't gives us much information. We're not much wiser. We know it's a complex disorder, and this image kind of confirms that. However, if we switch and we start looking at cell type specific pathways that are upregulated, the image changes. Now, this is mainly, you can see here on the left, it's dominated by this big purple box, which is RNA processing. This includes splicing, RNA binding, transport, etc. And this is globally upregulated in neurons in schizophrenic brains. Well, this is something that we're really interested in trying to follow up. This would suggest that maybe there are massive splicing differences because the machinery to perform the splicing is upregulated in patients. So together with a collaborator from Paula Geste Rodriguez in the University of Florida, we have performed long-read RNA sequencing on the same samples, not on the full samples, but on 10 cases and 15 controls. And doing so, we can identify genes that have differential splicing activity in patient and controls. And if we, this is their paper, but I'm going to show a little bit of data, which is basically if you look for what type of genes are being upregulated or not being differentially spliced, you can see that this is actually very overlapping what we find in the DEGs. So we find mainly mRNA binding, the spliceosomal complex itself. We have a lot of signal for synapses, and these in agreement, the major cell types are in agreement. We can also look for which cell types are enriched for splicing, and it is again the pyramidal cells. And also targets of RNA binding protein RBFOX 1 to 3 are enriched. This is splicing factors. So this is another point of convergence, which points towards the same cell types, even though we use bulk samples. It also points towards the same biological processes. So one thing that we're excited to follow up now, and I'm showing you very preliminary data, this is ongoing analysis in the lab, is to look at the spliceosome, the nuclear speckle, which is where splicing happens in the brain, in the cell. And you can see here that in schizophrenia samples, you can please ignore this large aggregation outside the nucleus, but you can look at, we're now looking into the structure of the nuclear speckles in cases and controls. And this is ongoing, and I'll happy to update you where we end up with this kind of data. Okay. Final piece is that we're now, of course, doing gene network analysis using an algorithm called HDWGCNA, and we can then find these gene modules, gene programs that are changing in neurons. And here's an example for layer 2-3 neurons, which are the main modules that are present in the neurons. And we see these different modules. These are now gene regulatory programs or bunch of genes that are co-regulated in the neuron. We do this on the entire data set. So we have to figure out, okay, this is now co-regulated modules. Which ones of those modules are now related to the disease or to disorder? And to do that, we can now look at in each of these modules that I have here, are they changed in patients versus control? And this is the plot down here. You can see that the red one is very it's a big log fold change cases to control, and is have a low p-value. So out of all these modules, it turns out that the red one is the one that is mostly related to disease. This is the most changed in patients. And so we can now do that for each of the cell types, and we get these bunch of modules that are disease related across the different cell types. They are now at this point, they're all cell type specific, right? We identify them separately in each cell type. So we can now ask the question, which ones are shared between cell types and which ones are specific to cell types. And what you can see here is that we plot now the % of gene overlap between the different modules, and you see that there is some structure here. There's a few modules here. One is that is shared between astrocytes, oligodendrocytes, inhibitor neurons, and excitatory neurons. Here's another one that is only in excitatory neurons. And then there's a few other down here that are shared between excitatory cells and oligodendrocytes, etc. So I'm showing you very early data. Basically, I got these plots a few days ago, so we're still in the process of trying to interpret them. So one of the things we want to do is, of course, to see look for which ones of these modules might be disease-causing and which ones might be related to the response to changes in the nervous system. So one interesting thing I just want to show you here is that actually the module that shows upregulation of mitochondria occurs in the same cells as the module that shows the upregulation of RNA processing. So this seems to be connected to each other, this big block here, even though they go in different directions. So this is something that we are further investigating now. Okay, so conclusion, a lot of different stories that I told you, but I wanted to make a few main kind of points here. One is that having quantitative measures of cellular diversity, that is, let's say, single cell resolution data, really opens new frontiers. What kind of questions can we ask of the nervous system? And I hope that I've convinced you that there is really a convergence of different modalities of investigation. We can do cell type and circuit genetic risk analysis, and that actually converges with the same kind of networks that we see using functional fMRI. And it confirms it's also the same cell types that are being implicated by post-mortem analysis. So I think this gives really hope for the field in that sense. Yes, it's a complex disorder, but there are definitely some common rules that several different types of investigation points towards the same. The big one here is clinical data. I hope to be able to integrate that at some point. Also, our findings that both genetic and fMRI data highlights the amygdala and hippocampal circuits. And I think that cell type specific analysis reveals disease biology, maybe not so surprising. This also points towards the same cell types that the genetic risk analysis did. The upper layer excitatory cell involvement is now a convergent finding, both between genetics and transcriptomics, but also between different studies. There are another published study and another that is on bioRxiv that points towards the same. So we're starting to see the same thing in different labs, which is really nice. I also told you that GWAS genes are generally down, while whole exome genes are upregulated in schizophrenia patients, which I think suggests maybe a model of stability where the brain is trying to stabilize itself, and maybe a lot of genetic risk is actually destabilizing mutations. Altered mRNA processing is really a hallmark of schizophrenia, and alternative splicing indicates the same process as we're seeing being differentially expressed. And what we're doing now is we're going further with the gene network analysis to really try to understand what that means. And we're also trying to use some of this data now to see whether we can achieve a first stab at patient stratification using molecular data. So with that, I'm happy to take questions. I want to first thank all the people that have been involved, both in my lab, but also my very long-term collaborator, Patrick Sullivan, Lu Yi, and people doing fMRI work, Johan Lundström and Joanna Volpe's lab, the people that have provided us with post-mortem data, and of course, a special thanks to the people that actually donate their brains post-mortem for us to do these kind of studies. And then a lot of work, our other collaborators. And with that, I'm happy to really take questions.
Thank you, Jens. That was really mind-blowing integration of so many levels and how that is enlightening our knowledge about so much that we don't know about this disease. The floor is now open for questions. Please type in your questions either in the chat box or raise your hand and we can unmute you. We can start with Pranchu, who has a question in the chat. Can we unmute her?
I can read it.
Okay.
So the question is, in the 106 dissection, how was the proximal dendrites or cytosolic content accounted for, assuming single nuclear RNA-seq encodes data set from isolated nuclei from these brains regions, especially eccentric MSNs? So this is a really good question because we've shown in a paper in 2018 that having nuclei data compared to whole cell data is very different in the gene set. So if anything, basically what we know is that by looking at the hippocampus, you have a very discrete pyramidal cell layer, and then you have a layer that is filled with dendrites and axon. So there was a paper where they dissected out the two and looked for things that were shipped out into dendrite and axons, mRNA that is shipped out dendrites and axon. And the proportion of mRNAs that are being actively exported out of the cell body is highly enriched for schizophrenia risk genes. It's really highly risked. And if you go and you take a single nuclear RNA-seq, you're, for some reason we don't really understand, depleting that signal. So you see less of it than if you have dissociate the whole cell in the mouse, you actually get a bit of proximal dendrite and stuff hanging onto the cell when you ship it to sequencing. If you take the nucleus, you lose that. So I would say that using the single nuclear RNA-seq, if anything, risks downplaying the role of neurons. We're missing neuronal specific signals that are very important. We don't account for proximal dendrites or cytosolic contents. That's the short answer. But this is we do this thinking that since we don't do it in any region, this might be fair. Yeah.
Hi, can I go ahead and ask my question, Ritika?
Sure, please.
Yeah, hi. Really nice talk, gents. It was wonderful, fantastic data and work. I had two questions. First was on your rare genetic variants. Mostly they are expressed actually throughout the brain. So there is no region-specific expression of some of these genes. So how do you think that, mutations in these are causing very specific amygdala or hippocampal circuitry defects? And how does that even explain, and additional question to that is, how will we achieve granularity on how any hippocampal or amygdala circuitry defects can lead to psychotic symptoms? So it is going from a very molecular to next circuitry and then next to behavior. That's where I want to see the connecting dots.
Yeah, I think that's it's a really great question. Experimentally, it's very hard because we don't know what a psychotic mouse looks like.
Okay.
We don't even know if they have psychosis. It's kind of hard to ask them, which is what we do with humans. So I don't think people have been trying to do endophenotypes. People say that they can have hallucinations. There's some papers saying that. I'm generally skeptical to that. So I don't know whether experimentally we will be able to address that, to be honest. if we're lucky, we can find genetic signal. Maybe there's a difference, and different patients have more signal in the amygdala. For the common variants, they are not evenly spread, right? That's what our work show. Our common variants are very specific to different regions and different cell types. The rare mutation, the rare variants are common. I agree with you. I don't think there's a large scale like effort now trying to use that we have these large effect size rare mutations. I think that our data, what it really shows is that these are mainly protective protective agents. That means that if you are modeling them in a knockout mouse, you are not looking at schizophrenia risk. You're not looking at schizophrenia risk or schizophrenia mechanisms. What you're looking at, my take on this is that you're looking at stability metrics. These genes are necessary to be there to stabilize the brain. If you can't upregulate them, you have destabilized the system. But it doesn't tell you about the initial insult, or it might not. So we'll have to see how this plays out, what these data show. But there is a significant risk, I think, that you are basically destabilizing the brain, but without having the correct push towards the schizophrenic state. Maybe maybe we're looking at something else. And in mouse models, we don't even know what that would look like. So I think that yeah, this is a very hard problem I mean, if you have an idea, I would love to talk to you about it.
I mean, perhaps I wondered, I mean, just to give a background, I'm Bhavna, and actually I'm a faculty at the Institute for Stem Cell Science, and we collaborate with the Center for, I mean, we use a resource from the CBM. And that's something we've discussed quite often, that should these studies also go with perhaps more in-depth fMRI studies to look at, are specific circuitries affected when they have these psychotic episodes, or some such thing.
I think so too. So the thing that I'm the most excited about is these what do you call it, at-risk patients. So I would love to have a cohort where you have people that have a high genetic risk that are there's different criteria to put them into high risk population, right? But to follow them and then see which ones do get psychosis, and are the ones that do get psychosis, which one develops schizophrenia.
Correct.
So this, I think, and if we can, fMRI is hard to work with because it's so multidimensional. So you have a problem of overfitting and multiple comparison. If you could have more maybe hypothesis driven, you could look in a fewer areas, and then we could see, hey, maybe there's a relative difference in this. So I'm really excited by the fMRI field. I'm coming from a place where I've never really cared about the fMRI field. But I think it has to be done on a large scale. The problem, I think, with the field is it's been done in too low scale. So I think there needs to be multi-center studies, or ideally, someone has 1,000 patients at risk and put them in the same scanner with the same operators. Of course, but I do think that the problem there is, again, is similar to what we're suffering from in the molecular field, is that we have too low ends. So to find this, but I think that this is my hope. I don't think maybe we can circumvent. I don't think that animal models is the right answer. I don't think that iPSCs might be the right answer, because I think this is post-adolescent development probably. But I'm excited about doing human research in this sense. But this will require huge efforts, of course.
Yes. Okay, thank you so much.
Anna, can we unmute Anna? Or you could just respond since it's in the chat.
Yeah, so Anna, you're here. Do you want to read your question, or should I read it for you?
I was just so curious at the end of your talk, a great talk, thank you. You mentioned sort of trying to maybe start stratifying patients with molecular data. I'm curious about how you might think about beginning to do that.
Yeah, so there's a major problem to overcome, which is human diversity. And I want to start out with saying we have an N of 40 in each. It's very low. We're nowhere near capturing the diversity. So what we can do, of course, is we have a very high dimensional data set in terms of gene expression in different cell types in a particular brain area in the prefrontal cortex. But still, we can there look at now, say, like, are there modular difference? Is there a subtype of patients that has a different molecular phenotype than other patients? So that's very easy to find. The problem is then whether this is disease, like does it have relevance to the disorder? Like does it give, so, and here, this is why I'm harking on the clinical data, the fact that like these are really well-described samples, but even so, it's really hard, like a lot of the clinical information we have is like yes or no. So it would be super nice to have much more detailed clinical description of these patients, because ultimately we can find different, we can see that the patient clusters in different clusters when we look at the molecular expression data. But there's two main things. One is like if we took the healthy people, they also cluster. And if we see different clusters between the patients and we don't know whether it's disease related or if we just took a random 40 people across the world, they would cluster into different types if you do single cell. So that's a major kind of thing that we're up against and we can't solve really. But we can take a first stab. So when we do this, we see that there are actually different gene sets that differ between different class of patients. And then, of course, what we really would like to show is that this has something to do with, let's say, treatment resistance or age of onset or, you know, some kind of response to a medication. But I'm afraid that, you know, until we have larger data sets, it's going to be hand waving. So we're going to, I think we're attempting to do this to see whether something comes out. Would have very low confidence in it, but at least we can explore this like methodologically and to see if there's anything there. We can also cluster them on, you know, basically together with PRS. Do they have high risk score for this or not? And comorbidities. It's too early days. We see clear patient clustering, but whether that is a real thing or not is what we're trying to convince ourselves over right now. Let's say that.
Thank you. Thanks.
Hi, I'm Meera Purushottam. I work with Biju and Ritika in the lab. So I put my question there. You mentioned splicing defects. Do you see splicing defects in nuclei, or does one have to use the cellular transcriptome? That was one question. And the other is a more philosophical question about those recent paper where they were talking about all post-mortem brains being different. And when right now we are, you know, talking about really going into great depths of looking at post-mortem data. So this is a kind of an upsetting paper. So I just wanted to ask you, what was your opinion on that?
I'll answer the first one, then I need a clarification on the second. The splicing defects, we can capture it using only nuclear data, but the signal is weaker. So the splicing happens in the nucleus. So you can look in the nucleus for unspliced version and spliced versions. You will find both of them in the nucleus. Of course, you will find much more of them if you look in the cytosol, right? Because that's a more over time integrated signal. But the acute situation you can look at in the nucleus. But the data that I showed you was on bulk. So this is basically taking a piece of tissue and looking at everything. But that includes all the cell types, etc. So we're moving towards this thing called single cell splicing analysis, where you can take a single cell and look at the splicing events happening in each cell. And then it's a very sparse signal. You have to aggregate many cells together, but you can then look at different things. So that's one direction that my lab is currently pursuing. With the report that post-mortem brains are different by themselves, you mean that the post-mortem process changes expression? Is that what you're? Yeah. That's probably true. I mean, I find it hard that the process of dying and would not change the gene expression would be strange to me. What I can say is that we don't see any big effects of post-mortem interval, or we don't see any large, like these things. We have a lot of batch effects between different patients or samples. And we have also, if we batch them together, we have two batch effects, then we have batch effects as well. But when we try to correct for all the known confounders that we have from the clinical data, which is like toxicology, post-mortem time, cause of death, none of those increases the power of our data. So I think it's we still don't know, but we don't see big effects of these things. We are like using random variables is better than known variables in terms of normalization. The one big difference is smoking. Basically, if you have nicotine in the blood, we can find very, then that makes a difference in gene expression. But that's it. Otherwise, we have better luck with random variables than we have with the known.
And have you done any correlation with the treatment that a patient may have received over life?
Yeah, we have information on treatment. It's a bit sparse in our samples, but we have information, not lifetime information. We have toxicology reports, so we know what was in the blood at the time of sampling. We also have a medical history on a few of them, like which type of medication they have been on, whether they followed that medication regime or not, we don't know. So we don't have a lifetime history, and this is one of the problems, I think. And also, like many of these patients have had very hard lives. Some of them, have taken other drugs and they have lived homeless, and there's a lot of things that have affected these people. So that's probably a cause of variability. And again, there's no, with 40, N of 40, it's too small to say. We don't know what is individual variability or what is variability induced by a different lifestyle or a different medication regime. There are studies out there that are trying to address this, and we are integrating that in our data in the analysis. I haven't showed it today, but basically they have given antipsychotics to non-human primates and measured the transcriptomic response. They've also looked at transcriptomic differences of patients on medication versus people not on medication. So there are these data sets where we can use to see, like, out of the signal that we see, how much of that is likely attributable to like drug effects of antipsychotic drugs. So that I didn't show that now because the data is a little bit, we've done a preliminary analysis, but we're trying to do this in a very careful manner. But yeah, again, with too small data set and too sparse clinical data, it's really hard to do it within your data yourself. So we rely now on other data sets that and try to clean our data based on those data sets. But
right. I just have one more last question. You talked about MRI initially in your talk. So do you have any data at all where you have both MRI and post-mortem? I know it's probably asking for too much, but
that would be great. If anyone asked that, I would be more than happy to analyze them for you. But no, this is a completely independent cohorts. And but what I really I'm really excited about is this cross ancestrally analysis. I think that it's intriguing that the incidence of schizophrenia is so similar across genetic ancestries, and I think that and if we could get fMRI data and molecular data from differential ancestries, I think much would be won. If we can try to get an understanding of how different is the baseline and how different is the disease signal, I think a lot could be won in terms of trying to understand the mechanisms behind the disorder, molecular mechanisms. So that is something that I'm super eager to get that type of analysis going.
Sure. Thank you.
Any more questions? Okay. Thank you so much for everyone for joining us today. On behalf of my co-organizers, I'd like to thank Jens for giving us the time and talking to us today, and for all of you for joining us tonight. And this, like I said at the beginning, is an event that we will be doing on a continuous basis. So do in the chat box, we have shared our links to keep in touch with us for our future events, both scientific presentations as well as public outreach events that we do as part of our regular events that we organize. Join us through your favorite social media channel, and we hope to see you again soon. Thank you.
Bye-bye, everyone. Thank you, guys. I'll just stay on here. Yeah. lowering my desk. Okay.